बंदे हैं हम उसके, हम पे किसका जोर। उम्मीदों के सूरज निकले चारों ओर। इरादे हैं फौलादी, हिम्मती हर कदम, अपने हाथों किस्मत लिखने आज चले हम। Welcome everybody to this special broadcast, a special session in the remembrance of a special person. Welcome to this webinar, guys, and I'm your host, Shekhar Suman, and it gives me immense pleasure to be a part of this session. You know, we all are not human beings. We all are divine beings undergoing a human experience. And today, as I come to you live, I want to share a few words about this divine being who changed the way things happen around him. He is not just a legend or enigma. He has been a leader throughout his life and career. Today, Sri Sudarshan L. Gowda stands in front of us, tall, with all his achievements. And that's the reason today we have a very special person, his daughter, Ms Deepthi, presenting her achievements with all of you, sharing her PhD work with all of you. And we all will join hands to remember him and his achievements. We have a long list of guests who would be joining us shortly and talking about him and his achievements. But what is going to be a special attraction for all of you today is a daughter who walked on the footsteps of her father. You know, behind every great daughter, there is an amazing father. And late Sri Sudarshan L. Gowda was a very responsible and amazing father, and the result you will see today. Ms. Deepthi is right now finishing her PhD work in Montreal, Canada, and she'll be talking about today's topic, which is how do chromosomal translocations promote cancer. That's her PhD work. And I'm, as always, joined by my mentor, my guru, Dr. Jagdish Mittur. And I would like to invite him on stage, please. Dr. Mittur, if you could join us. Welcome, Dr. Mittur, and thank you so much for joining us. Over to you. If you would like to share some words about late Sri Sudarshan Gowda ji.
Thank you, Shekhar. Thank you for hosting this. This is a special event for all of us, those of us who knew Sudarshan L. Gowda and his family both professionally as well as privately as families. I just want to take about maybe 5 minutes to share with you all my association with Dr. Sudarshan L. Gowda. You know, the association of Mr. Sudarshan Gowda's family and my family goes back to the times when my own parents and Sudarshan's parents were neighbors during the times when my father used to work for the PWD Department of Government of Karnataka in Sakleshpur, a lovely place in Karnataka. This was well before I was born to my parents. Subsequently, and several years later, when my parents relocated to Bangalore, more children were born, and I guess after a lot, I was one of them. Right from my young age, I used to hear the names of Sudarshan's father, Mr. Lakshmipati Gowda, and other members of their family. Once, I even had the opportunity to meet Mr. Lakshmipati Gowda as a young boy. He was tall and a lean gentleman, and I remember being astonished by his height. Well, several decades later, when I had joined Monsanto Research Center as a leader of a group, and in a rapid spree of hiring a bunch of smart scientists and technologists, there walked into the interview room Dr. Padmini Sudarshan seeking a job. Not knowing who she actually was, Padmini was hired for the Arabidopsis plant data mining project, along with many others, and she became a leader of one of the sub-teams in my large team. Only days later, during a lunchtime casual conversation, I came to know that she is the daughter-in-law of Mr. Lakshmipati Gowda and wife of Dr. Sudarshan L. Gowda. I was so very happy, of course, as destiny had brought the children of two families who were neighbors in Sakleshpur now together in Bangalore. Of course, our respective parents were neighbors, I guess, in heaven by then. And a few months later, at a lunchtime gathering of my team in our rented home in 16th Cross, Malleshwaram, which was open to families also because it was lunchtime, there walked in Sudarshan. He actually arrived a bit late, and that too, at a time Padmini, not knowing that he was coming, was singing a very appropriate Hindi song from the year 1966 movie Suraj. Baharon Fulu Barsao, Mera Mehboob Aya Hai. I'm sorry, my singing is not so good. But you know the song, a very famous song. It was then that I actually met Mr. Sudarshan A. Gowda, although I had seen his father many, many decades ago. I must tell you, Sudarshan was a replica of my image as a young boy of his father, Mr. Lakshmipati Gowda. There was endless teasing, of course, of the couple Padmini and Sudarshan as Padmini was ending her song from all my team members, and it was a lot of fun. I still remember that afternoon very vividly. Now, I would like to switch quickly to the career of Mr. Sudarshan L. Gowda. Sudarshan was born the youngest of 7 siblings in 1957 in Gupi, Tumkur. Sudarshan joined pre-university at National College. He further pursued his undergraduation, specializing in fisheries science at the University of Agricultural Sciences, Mangalore campus, securing a gold medal. After obtaining a degree in MSc in biochemistry, he soon joined as a faculty at the University of Agricultural Sciences, Bangalore, the GKVK campus. Sudarshan dearly loved teaching as a profession, much to the advantage of his students. Sudarshan was beginning to blossom in his life academically and as an ardent family man. Unfortunately for all of us on the planet Earth, the forces from the heavens were very powerful and unfortunately took him away at a very young age. Sudarshan passed away in December 2002, leaving behind a legacy as a responsible son, a loving husband, and a passionate teacher, and a very, very loving father, as well as a great colleague and a sincere friend to so many of us. I must mention here that Sudarshan's family, led by his wife, Dr. Padmini Sudarshan, has institutionalized a medal called Sudarshan Memorial Gold Medal to be awarded each year to a high merit student who has excelled in the field of biochemistry, the favorite subject of late Sudarshan. Thank you so much, Padmini and family, for institutionalizing Sudarshan's name in that. And this year, I think a young girl from Tumkur district, I think, she won that Sudarshan, the coveted Sudarshan Memorial Gold Medal. I'm sure she will remember his name and the family throughout her life as she progresses in her own career. What is amazing to me is the commitment and determination of his family, that is Padmini and 2 daughters, Deepthi and Kirtana, to move on. Absolutely inspiring. Thanks to Padmini and her daughters to show the rest the way to move forward. The determination of a young and lovely Deepthi, who was only 13 years young at that time of her father leaving her to pursue a career that her dear father had pursued, is highly inspirational. That was our way of remembering him forever. Of course, the younger 8-year-old sister Kirtana, who pursued music, which to me is a symbol of what Padmini was singing at the time when Sudarshan walked into our home back in 1999. Well, the story, while it is sad, is an extremely inspiring one for anyone to show that we must move on towards making the world a better place despite these setbacks in our lives. And that to a scientist, we must make a difference. Thank you so much, Shekhar. I might have taken extra time, so let's please move on to the next item. Thank you so much.
Hello, everyone. Good evening, good morning, depending on where you are dialing in from. It's my pleasure and privilege to introduce Deepthi Sudarshan today. My association with Deepthi is really over 2 decades, right when she was a middle schooler. And it is indeed a very proud moment for me to introduce Deepthi today. She has a very impressive research and academic career and is a role model for many students and a mentor for aspiring young researchers. Currently, Deepthi is a doctoral student of cell and molecular biology in St. Patrick's Cancer Center at University of Laval at Quebec City, Canada. with research focus on chromosomal translocations in sarcomas. She is recipient of scholarship for doctoral training from FRQS, and also a recipient of FRQNT, Merit Scholarship Program for foreign students for PhD studies. In addition to this, she has won many prestigious awards, travel grants for her posters and presentations at international conferences. Before pursuing PhD, Deepthi was a research assistant at Professor Kundu's lab in Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, India, where she worked on chromatin dynamics and transcription regulation, focusing mainly on the role of histone chaperones in transcriptional regulation. Deepthi has already co-authored 2 book chapters, published 4 articles in peer-reviewed journals, and a few more are in preprint version. And she also has a patent application to her credit. Publications are mainly focusing on the oncogene expressions and histone chaperones mediated transcription of oncogenes. So everything around expression of oncogenes. Deepthi completed a post-graduation in biochemical genetics from Vellore Institute of Technology. While at Vellore, she received merit scholarship for best academic performance and also was a silver medalist with 2nd rank. She did her undergraduation from Mount Carmel College with a major in chemistry, zoology, microbiology, with a distinction and certificate of excellence in pretty much every subject that she majored, right? Chemistry, zoology, and she was very active in student committees and science and educational outreach activities. This is only a trailer of Deepthi's academic achievements. The list continues to be very impressive. On a personal front, as an individual, Deepthi is very caring, calm, considerate, and also a very confident young lady. She's a 10 on 10 daughter and a great friend and like a second mom to her sister Kirtana. When Deepthi was just 13 years old, she was taking care of her younger sister Kirtana, who was also just 8-year-old when, mom was at work. So Deepthi is also a bundle of talent, great artist. She draws well, paints awesome, super creative, sings very well. All the genes from mom has gone to Deepthi and Kirtana in being creative and so very talented. And we as a family, have been on many trips and treks together, and she's very fun-loving and adventurous too. In addition, she has a very balanced, practical view of situations, circumstances, and guides very appropriately, not just for her peer groups, but even grown-up adults like me. And this has made Deepthi as a go-to friend for many. Deepthi is married to Dr. Samarth, a bioinformatician and a very fine gentleman. And, he is mother-in-law's favorite too. Today, Deepthi has generously agreed to share with us her research findings in the area of oncogene expression. And I'm sure all of you agree with me that a breakthrough understanding in this domain of science is really the need of the hour, with sarcoma of various types increasing across the globe. So with that, Deepthi, we are really eager to listen to you today. Once again, thank you for taking time to share your research highlights with us. Over to you, Deepthi. Thank you.
Deepthi, sorry to interrupt. Kindly unmute your mic.
Can you hear me now?
Yes, Deepthi. Thank you.
Yeah, sorry, I had shared my screen and I couldn't really see anything. Okay, I just wanted to say thanks to everybody who organized this event, Jagdish Uncle, Biotechnica platform. Thank you, Vidya Aunty, for introducing me. I'm going to share my screen and start the presentation now. Okay. So yeah, first things first. I cannot really say too much about my father without getting emotional, so I'm going to keep it short. I think I really learned a lot of things in childhood from my father, and usually you don't realize the lessons that are being taught to you when you're a child. But looking back, I feel like a lot of the things that have helped me as an adult, not only in my career but also in my personal life, were taught to me very well through both of my parents. And I think the biggest thing from my father is curiosity. I think as a child, just being around him, I learned to be curious and patient about learning. I struggled with learning certain subjects, I think especially math and science. And my father used to sit patiently with me and try to simplify the concepts, and magically I understood everything. So that taught me that you need appreciation, curiosity, and a little bit of patience to learn things. And once you do learn things, you see the world in a different way. I also remember being being in GKVK. That is very fond memory. So being in the lab where he was working, so I remember agarose gels and going into the cold room, and probably not the best for a child, but these are really precious memories. And also being in his classes while he was teaching. So in a way, I was his student too, I think. And I, as a child, I didn't know about his successes and his achievements, and it's only hearing from other people that I get to know. And I'm really thankful to everybody who knew him, who convey these things to me. Especially all the students of his that I meet. They tell me about how great of a teacher he was. He used to always listen to students and take the effort to teach them, even after class hours. He was always there to help. This is really wonderful for me to hear, and obviously these are very big shoes for me to fill. Um, I think I pursued science because I was inspired by him, and hopefully the science that I'm doing is um, well thought out and interesting and useful enough to impress him, I guess. So I'm going to start my presentation about my work. Yeah, so I will try to be as simple and clear about the concepts in the beginning. I don't think I'll be going into too much detail about my research. If interested, of course, I am available to chat about my research. So to first begin, we know that all living organisms are made up of cells. I'm just gonna get my pointer. Yeah. So all living organisms are made up of cells, and cells, you can think of cells as kind of the basic working unit inside the body. And the cells have a very important a molecule called DNA. So DNA is our genetic information. So it is like a blueprint for producing everything inside our cell, and also for the functioning of the cell and ultimately the entire organism. The problem here is DNA. The amount of information that has to be stored in the DNA for a particular organism is usually very large. So the DNA molecule is actually really large in terms of length. For example, if you take all the DNA inside one cell, it should be around 2.5 meters long. So since our cells are quite tiny and it has to be fit inside this cellular component called nucleus, it has to be quite condensed, otherwise it cannot be fit into the cell. So that's where chromosomes come in. So chromosomes are basically molecules of DNA. So chromosome 1 is one molecule of DNA, 2 another molecule of DNA, something like that. And they're quite long, and they look like this because it is condensed. It is condensed quite a bit to form very compact structure. And this chromosomal structure actually is quite useful when the cells have to divide. So this makes it easy for the cell to kind of replicate the DNA, and when the cells are dividing, it can transfer one copy of the DNA to another cell without it getting jumbled up with all the other material inside the nucleus. So this is basically a karyotype, or just an image of how the chromosomes look like in the human cell. So all of us have 23 molecules of chromosomes, and if you're a male or a female, depending on that, you either have XY or XX chromosomes. So I was talking to you about DNA. So I told you that DNA is a genetic material, and it carries all the information that is needed. So how is this information carried? This information is carried actually in genes. Right now, there's also information, there's also research on all the extragenic material in the DNA, also leading to some functions. But right now, we will focus on genes. Genes are basically segments within the DNA that encode particular information to make a protein. So for example, here is a stretch of DNA. This is in fact quite short, but this is how it usually looks like. It has a double strand, and it has these nucleotides A, T, G, and C, which arranged in a particular format encodes some information. And this information inside the DNA in a particular stretch called a gene can be transcribed into something called messenger RNA. So transcription actually takes the information in DNA and converts it into a message that is similar to DNA but slightly different. And this is a single-stranded molecule called mRNA. I'm sure you have all heard of mRNA, since this is the material that is used in the recent COVID vaccines. So messenger RNAs are actually molecules that carry the information to encode molecules called proteins. So for example, here, 3 nucleotides here, UAC, mean that the translational machinery, or the molecules that are making the translation happen, to encode for a amino acid called tyrosine. And then there is a long stretch of amino acid which fold into a molecule called proteins. And proteins are basically the workers in the cell. These are the material that make up the structure and also carry out all the functions inside the cells, and hence build your flesh, blood, muscles, etc. So coming to the topic today, what are chromosomal translocations? So we understood what chromosomes are right now, and usually in a healthy human cell, all the chromosomes are intact, and they kind of look like this cartoon. But sometimes DNA damage can happen. So the sources of DNA damage cannot just be external factors like ionizing radiation, smoking, UV exposure, etc. These are all sources of DNA damage, but it can also be internal factors of just the metabolism inside the cell. Recombination events, oxidative stress, etc., can cause DNA damage. That means there is a break in the DNA molecule, and this is not good. So usually, if everything is going well, the cell 99% of the time repairs this DNA damage and it goes back to normal. But sometimes it happens that this damage is not corrected well, and many things can happen. For example, a part of the chromosome can be lost, so there's a deletion of this chromosome. If there is two sites of DNA damage, material can get flipped. So you remember that I told you that the information coded in the DNA is important and the directionality is also important. So it needs to go from one direction to another, but sometimes when it gets flipped, there is a problem. And the other thing that can happen are translocations. So if there are two chromosomes and both of them have breaks, it could happen that the chromosomal material can get flipped. So the machinery that is repairing DNA damage has joined these two segments, but they're not supposed to go together. And likewise here. So since the DNA molecule from here is combined to a DNA molecule from another chromosome, it is not in the right orientation and it's not next to what it's supposed to be. So that can cause problems. So this is a chromosomal translocation. And chromosomal translocations are pretty frequent in certain types of cancers, especially blood cancers and sarcomas. They occur quite frequently. And they're usually kind of the drivers of these cancers. That means they tend to be the main factors for a cancer to develop. So what happens when there is a translocation? So when there's a translocation, the region of a gene which is supposed to make a protein can come under the control of particular segments of DNA that is not supposed to be there. And this can drive the gene to be expressed a lot more. So instead of being produced in a certain amount, it gets produced maybe double or triple the amount that is supposed to be produced. And this can cause problems because all the proteins need to be balanced. It needs to be in a homeostatic way in the cell, and overexpression of a particular protein may cause problems. Another thing that can happen is that half of a gene might be here and half of a gene might be in the other segment, and both of them can come next to each other and form almost like a mutant chimeric protein. And this can cause problems because proteins actually have every region of the protein has a particular function. For example, the beginning of the protein can have some function to direct it inside the cell, where to go or what to bind, etc. And when different regions of different proteins come together, they form a completely different function from what individually they were supposed to do. And that can be problematic to the cell and it might lead to disease conditions. Another thing that can happen is the gene can get disrupted. For example, it could get cut off, or it could not be processed properly because it is now sitting in another region in another chromosome, and could lead to the loss of function of this protein. So basically, the protein is not produced, and the lack of this protein can lead to problems in the cell. So all of these events occur randomly. It's not like translocation occurs in a particular place for it to drive the cancer. It's not like the cell kind of knows what to mutate. So it occurs randomly, and when something like a random event like this happens, and it creates like a protein that is chimeric and it is giving some sort of growth advantage to the cell, then that gets selected because it is giving the cell an advantage to keep growing or not die or whatever, and that gets retained in the cell, and that's how they act as kind of drivers of particular cancers. So how do they actually cause cancer? Yeah, I will come to that in a minute. Just to recap what I said. So this is basically just a comparison between what would be normal and what would happen when there is a translocation. So for example, here there are two chromosomes illustrated. There is gene on one and the gene on another chromosome. And originally, this gene would be expressed and another gene would be expressed, and they would form two different proteins, and there's normal growth. In the case of DNA damage and chromosomal translocation, the regions of the chromosome gets rearranged, and you could end up with a situation where you're creating a fusion of genes A and B, and this could lead to the production of a fused protein, and this is probably not really good, a good thing. It could lead to proliferation of the cell to a bad extent, and may lead to a condition like cancer. So most of these events, like mutations or deletions or whatever, if they're leading to cancer, they impinge on any one of these hallmarks. There are of course more, but I'm illustrating the most well-studied ones here. So for example, formation of a fusion protein could give a cell an advantage by, for example, resisting cell death. So there is a signal for a cell when it is when there is enough DNA damage, or there is accumulated oxidative stress for it to die. But if there is a fusion protein that has formed inside the cell, it could give the cell an advantage by not dying. It could resist any of the signals coming saying it's time to die. The cell might not die and just keep proliferating, and this could lead to cancer. So any of these things illustrated could lead to cancer. And since chromosomal translocations are not uniform, like not all chromosomal translocation have the same way of causing cancer. You all might have heard that cancer is not one disease, it's basically a million disease. Even in one tissue, depending on the kind of genetic abnormalities that happen, different kinds of cancers can occur, and they all have different mechanisms. So in the same way, chromosomal translocations are not all the same. Different fusion of different genes can lead to different outcomes, but they impinge on any one of the mechanism to finally lead to cancer. So coming to what I am really studying right now in my PhD, the lab that I work in is very interested in something called chromatin, and the structure and function of this chromatin. So what is chromatin? I explained to you about chromosome and the problem of DNA being very long to fit inside the nucleus of the cell. So this is basically a continuation of that. So chromosomes are a really, really condensed state, and that only happens during cell division. But when the cells are just performing its normal activities and not really wanting to divide, the DNA is in a slightly more relaxed format, and that is called chromatin. And chromatin is also condensed because DNA cannot just be left just like that inside the nucleus. It needs to be organized and stored efficiently. And another problem with DNA is that all the cells in our body have the same DNA, right? Like everything from our eyes to our skin all have the same DNA, but every cell, like every tissue, needs to perform a different function. So gene expression, or the kind of genes that are expressed in our eyes compared to our skin, is quite different. And this is achieved by regulating what genes are actually expressed. So this regulation actually occurs through a kind of a structural way. So the raw DNA in our nucleus is actually wrapped around proteins called histones to form something called a nucleosome. So basically our DNA is wrapped around this kind of like a ball structure here that is illustrated, called nucleosome, twice, and then it is wound further into this structure and stored inside the cell. And the amazing thing about this structure is that it's not it's not just like this. It's not just stored like this. It can move and it can be rearranged depending on what function needs to be done. So, for example, a particular gene on this stretch, let's say, needs to be expressed because we need that protein. Nucleosome that are here can be moved around so that this gene is accessible for the transcription machinery, for example, to produce mRNA and then to produce proteins. The same thing can be said for example, DNA damage, repair, etc. So basically, a particular stretch of DNA needs to be exposed at a particular time for proper functioning of different cells. And this is achieved by regulation of the proteins around which the DNA is wound around. So this nucleosomes that I'm talking about are made of proteins, and these proteins have different domains. So the protein histones here actually have something called tails. So they actually kind of look like tails. They are protruding structure that sit outside of this nucleosome structure, and they act as platforms to produce to actually retain certain chemical modifications called post-translational modifications. So these are what you can think of these as flags. So basically, depending on what's happening in the cell, certain enzymes can put tags or flags on the nucleosome saying this is the right time to, let's say, move away from this stretch of DNA, or to come closer and condense this particular DNA, etc. So these tags that I'm talking to you about are called histone modification or post-translational modification. And two of the different tags that I'll be talking to you today are methylation and acetylation. So basically, there are two different chemical modifications that can be added, and both of them have different meanings. Based on where they are on the histone tails. So for example, you see all these histone tails here, and different amino acids here have different modifications illustrated. For example, K5 on histone H2B is acetylated, can mean something, whereas on histone H3, K27 methylation can mean something different. And how is this modifications, how are they actually written, or how are they actually recognized, and how do they bring about this function? For example, in transcription is what we are studying. It is actually quite well described. For example, I was talking to you about transcription. Let's say this particular stretch of DNA has a gene, and that needs to be transcribed because that's the right time for the cell to have that protein. Then there are particular histone modifications that can occur on the nucleosome that is kind of wrapping this stretch of DNA. And this gives it a signal that it needs to kind of move away and open up this stretch of DNA. And then other proteins that can perform this transcription process. These are RNA polymerases. These are big protein complex machinery. They come and bind here and they produce RNA. And once the transcription is done, there is again a signal for it to go back to being condensed, and that gene is not going to be expressed anymore. So all of these functions are actually brought about by proteins. Like I said, proteins are the ones that basically orchestrate everything. So here also these post-translational modifications and this moving away of nucleosome, all of this is brought about by different proteins. So there are certain classes of proteins or enzymes that can write histone modification. Basically, they can put the histone modification on the histone tail. These we call writers. An example of writers that I'll be talking to you today about are histone acetyl transferases, or HATs. These acetyl transferases, as the name suggests, add acetyl groups to histone tails, and histone methyl transferases, or HMTs, add methyl groups to histone tails. There are also proteins that can take away these modifications once they're not required there. And importantly, there are proteins that have pockets, sort of, to recognize a particular modification, for example, acetylation, and then tell the cellular machinery, okay, this is the place to do a certain thing. And these different proteins don't act alone. They are usually part of multi-subunit complexes. So basically, you can think of them as machines. So like in a machine, every different part does a particular function, and all of them together can do a task. In the same way, different proteins come together to form this complex, and each of them might have very modular different function. For example, there could be a reader that can read a particular histone modification, and it could then couple that to a protein that can erase a modification, or it could couple that to a reader that can add a particular modification and bring about a particular function in a stretch of DNA at a particular time. So this enables like time specific and place specific activity of all of these enzymes, and they really are kind of cellular machinery. So for example, what am I talking about when I say acetylation and methylation? So there is a complex called histone acetyltransferase that can add this acetylation mark to a histone protein on a nucleosome, and this acetylation mark can lead to opening up of the chromatin and, for example, transcription to be turned on. There are histone methyltransferases, like I said before, that can add this methylation mark on histones, and this could be a mark of condensed chromatin. That means the nucleosomes are quite close together, the DNA is more compacted, and transcription cannot happen here. So the gene expression is turned off for this particular stretch of DNA. So coming to really the crux of this talk, chromosomal translocations of proteins, of these chromatin proteins. So that's what I study. So there are a lot of translocations. When I say recurrent translocation, I mean that the same translocation or the same fusion gene is seen in multiple patients of a particular cancer. And this particular recurrent translocation that I'm studying actually fuse chromatin proteins. So the proteins that I talked to you about right now, those proteins that can add histone modifications or read histone modification, these are chromatin proteins. And when translocation happens and these kind of proteins are fused, what happens? Because these are the ones that are regulating the cellular processes. So what happens when these regulators get jumbled up? So the recurrent translocations that I am interested in actually occur in these sarcomas: endometrial stromal sarcoma, ossifying fibromyxoid tumors, and cardiac ossifying sarcomas. These are all different types of sarcomas. Sarcomas are actually connective tissue cancers. They're quite rare. And they also are known to have been driven by chromosomal translocations. So these are all the list of different fusion proteins that happen because of translocation, and these are all recurrent. That means multiple patients show these different fusion proteins. And the fusion protein that I am working on is EPC PHF1 fusion protein. Like you can see that it is recurrent in this sarcoma as well as this sarcoma. And this is what the protein structure can be illustrated as. So all of these different colors are different domains of the proteins which have particular functions. So this fusion protein fuses this part of the EPC1 protein to almost whole of the PHF1 protein. So this EPC1 protein is actually part of this TIP60 complex. Like I told you before, some of these proteins do not act alone. They are always in a protein complex to do a particular function. So it's important to understand what they're part of to really know what the protein is doing. So this EPC1 is part of this TIP60 complex, and it is actually, this complex is involved in transcriptional activation, and it's a histone acetyltransferase. What I told you before, it can add acetyl groups to histones and lead to activation of transcription. Whereas PHF1 is actually part of polycomb repressive complex 2, as you can see here. And this complex is actually involved in transcriptional repression, and it is a histone methyltransferase. So basically, this fusion protein is fusing part of proteins from an transcriptional activating complex to a transcriptional repressive complex. So this is quite interesting what can happen when two completely diametrically opposite complexes are being fused together. So I will talk to you about one of my experiments in trying to understand the molecular mechanism of this fusion protein. Let me get into the nitty-gritty of the experiment. I am explaining a biochemical experiment here, just because my dad was really interested in biochemistry. So what I'm trying to do here is to understand what this EPC-PHF1 fusion protein is interacting with. So the nucleus has a lot of proteins, and only particular proteins interact with each other to actually become a protein complex. And I want to know what this EPC-PHF1 is interacting with, so that I can understand its function. And what I'm trying to do is basically I have my fusion protein, and then I have I engineer it to have extra proteins that can form particular domains, and these are called tags. These are particular stretches of proteins that can be identified by antibodies that we have produced. So basically, it's a toolkit that we use. So we have a particular stretch of protein that we know, and we immunize that into rabbit or mouse or whatever, and produce antibodies against it. And we can use these antibodies to recognize the protein that is next to that tag. So I'm basically using the tag next to my fusion protein so that I can recognize the fusion protein. And what I'm going to do is break the cells apart, get a mixture of different proteins. Then I use this antibody against these tags to kind of pull down only the protein that is interesting to me. And I do it a second time. And then I get only the protein complex that my protein of interest is interacting with. So I use cells that have this EPC-PHF1 fusion protein with a tag, and then I crush it, and then I make extracts, and then I do purifications. So this is what is illustrated here. So I take like cells and I grow it in a bulk. I don't know if this is going to work. Yeah. So yeah, this is how growing, I think this is 2 liters of cell looks like. So we grow cells to like really large quantities. And then we take only the cells in the end, pellet it down, and then use basically a grinder to grind it and get an extract. And then we do what I described to you before, a purification. So first I use one tag to purify as a first step, and then I use another tag to purify a second time, and then I use this purified complex to do biochemical assays. For example, I want to visualize what all I got, so this I can do by actually staining a gel after I run it. If I have antibodies against particular proteins, I can do Western blot. I can actually identify each of the proteins through mass spectrometry. So this is a technique where proteins can be identified based on their particular mass. And we can also do structural studies, such as under cryo-electron microscopy, X-ray diffusion, etc. Yeah, so what was the result of this experiment? When I purified this EPC PHF1 protein, when I ran it on a gel, this is how it looks like. So obviously you can identify here that there are many, many bands, and each of the band represents a different protein. So the EPC PHF1 protein is not alone. It is forming, it is coming down and interacting with a lot of other proteins. That is what you can appreciate from this image. And then we identified what are these proteins by mass spectrometry. And these are all the names of the different proteins that are interacting with the EPC PHF1. And if you classify what these proteins are, you can see here the green regions here. All of these proteins actually belong to the TIP60 complex, and all of the ones in purple here belong to the PRC 2.1 complex. And we talked about it before that the EPC1 and PHF1 are actually interacting with their respective complexes together. So the question was whether it would interact with just one complex, it would dissociate another complex, or it will not, like associate with any of them. But what we actually found is that this is not very common, that this fusion protein is able to interact with this complex that has a lot of proteins as well as this complex. So essentially forming a sort of a mega complex. So you can think of this fusion protein not acting just as a fusion protein, but acting as a fusion complex. So it's basically bringing two different machineries together to form a new kind of machinery. And this is what is probably causing the oncogenicity, or this is what is causing the cancer in these particular sarcomas. So we repeated this kind of experiment with two other fusion proteins found in the endometrial stromal sarcoma, Jazafan SUS12 as well as MBTT1-EZIP, and we found the same thing. It's basically merging this one activator complex to a repressor complex. So what we see from the literature and from our own study is that basically in endometrial stromal sarcomas, the oncogenic mechanism seems to be to physically merge this TIP60 complex to PRC2 complex through various fusion proteins. So all of these are part of the TIP60 complex. These are different proteins that are actually found in the TIP60 complex. And these seem to be fused to proteins such as these, which are part of the transcriptional repressive complexes to form these fusion proteins and ultimately fusion complexes. And of course, we did a lot more experiments, which I'm not going to go into the details about. I'm just going to give you like a brief summary of what we found. So I explained the first experiment to you, that EPC PHF1 seems to assemble a mega complex. And since this mega complex has two different kinds of machinery, and they actually go and occupy different regions of chromatin, what we found was that the fusion complex seems to be occupying regions that are supposed to be transcriptionally repressed. It means these genes were not supposed to be expressed. And then it is bringing the TIP60 complex, which is a hat, which causes transcriptional activation to these regions. So basically, it's bringing an activator to a region of the chromatin that is not supposed to be active. And what it's doing there is to increase transcriptional activation of those particular genes. And this increased transcriptional activation might be activating certain oncogenic or genes that promote cancer with different kinds of ways, like I talked to you about, maybe suppressing cell death or increasing cell cycle, etc. And that is what is causing this oncogenic transformation of these normal cells. And we see the same thing with other translocations found in endometrial stromal sarcoma. So that is the project right now. It's almost a complete project. We have the non-peer-reviewed version of this project up on bioRxiv. If you're really interested to read through the project, you can read this, and hopefully soon we are doing revision, so the actual peer-reviewed paper is going to come out in a couple of months. Of course, there are a lot of questions that weren't answered in my project. And these are the perspectives. Like, we don't really know what are these gene pathways that are changed in cancer, because we are studying this fusion protein in cell lines in the lab. We are not studying it within patient samples or within patient sample derived cell lines. And this is actually difficult to do because these cancers are very rare and it's difficult for us to get these samples. But recently we were able to get some samples from patients. So hopefully we can identify the gene pathways that are actually changed in the cancer, so that we know which pathway it's taking to cause cancer. And also, we are not a lab that works on therapeutics, but we are hoping that our basic molecular mechanism of how this fusion protein acts can aid in therapy down the line. So I want to acknowledge, obviously, the people that enabled me to do this project. My research director, Jacques Cote, all the people who are actually involved in the study. So the project has a lot of other people who contributed. Bioinformatic analysis and collaborators. And this is my contact if you want to write to me with questions, etc. And these are all the different funding agencies that have enabled this study. This is my lab, and this is Quebec City. Thank you for your patience. I once again want to thank everybody who organized this lecture, and hopefully we are able to continue this at least, I don't know, once in a few years. And I hope to be able to hear from other students of my dad and what they're doing right now. I also want to thank everybody who helped me to do this project. So my family, my mom, my sister, and Samarth, and my friends. And I'm ready to take any questions.
Thank you, Deepthi. That was very clear, very lucid, very articulated in a very nice and simple manner. I kept seeing comments from many of the listeners saying that it's so beautifully explained, so well explained in a very simple manner that even a non-biology student could understand at least most of it. Of course, you're getting into more and more complex aspects of the gene regulation, pathway manifestation, and so on. So it's not easy, but you are on the right track to really unfold the complexity of what causes cancer, and we all wish you the best, Deepthi. I think there are a few questions. I think Sweta or Shekhar, can you please bring up the questions, please? And others can actually start asking questions if there are any. Okay, first question is, how are chromosomal translocations detected? Are there highly refined cytogenetic methods for you and the researchers to detect it at the earliest, so that we can prevent initiation of the cancer and hopefully metastasis of the same thing?
Okay, so can I share my screen again? Hopefully I can. Yeah. Can you see this?
Screen is not visible yet.
Okay. That's fine. So yes, so there are refined cytogenetic methods. Actually, you nailed the problem on the head. Yes, so usually chromosomal translocations are identified after a tumor is formed, right? So when the doctor is operating and they want to know what this tumor is, why is it being formed, they take a section of the tumor and then do some cytogenetic analysis. For example, they can do karyotype, so they can visualize the chromosomes and see if they are intact. If the part of a chromosome is missing or it's found with another chromosome, then you know a chromosomal translocation has happened. Other ways is to actually, when you know, for example, for endometrial stromal sarcoma here. You know that the occurrence of fusion is quite common. In those cases, they go through a routine where they can even take antibodies against proteins such as JAZF1 and stain it and to see if it is kind of overexpressed. Then you can see. Nowadays, there's also a better way to do it, which is to do second generation sequencing, so NGS analysis. So you can sequence the tumor RNA, and then you can detect fusion. So there are algorithms that can detect fusions. That is a more precise way that can also help you, because translocations can lead to gene expression changes. And you can, if you do like a RNA sequencing, for example, you can detect the changes in the genes too, like what is the consequence of the translocation, as well as just finding the translocation. But about the question about can we prevent it by detecting it, I don't think so, because it's always like retroactively detected. So as far as I know, not yet, but now there is also circulating tumor DNA detection that can be done with blood. And apparently some of the fusion proteins are amenable to this kind of detection. So that is a growing emerging field right now, to be able to detect any of these kind of fused transcript as circular DNA in the blood, which could probably help us to detect it early and prevent cancer progression.
Thank you. I think one of the other questions that came up, just going to come up on the screen now. So you did mention that there are translocations can happen randomly owing to exposure to different environmental toxic agents. So how can you tell if a given fusion protein resulting from such a translocation is causing cancer or will cause cancer? Is there a quick way to know that and to save initiation of cancer and metastasis?
Yeah, so we don't know if a fusion protein will occur, right? Like there are certain prerequisites for mutations to occur. For example, exposure to certain things is of course the first thing, but also, even if you have never been exposed to any of these things, just the fact that we've been able to live for longer and our machinery is working for a longer time, and it's prone to problems, itself can accumulate certain genetic damages and stuff, and it can lead to formation of these fusion proteins or etc. And how do we know that this fusion protein is causing cancer? This is a good question, because usually in cancer, it's never one thing, it's an accumulation of things. So there is a two-hit hypothesis in cancer. So basically you need one mutation plus another mutation for it to accumulate enough damage for it to lead to cancer. So this is probably the same thing for fusion protein. But usually the cancer that are driven by fusion proteins actually have very simple karyotype. That means other things are not really happening. It's just this fusion and maybe 2 or 3 other mutation that are usually detected in patient samples, and that is what is driving this cancer, and that's what makes it very useful to study these fusion protein driven cancers, because probably we can come to a clear understanding of what is happening. Because in other cancer, usually the mutations and the problems in the genetic material is so much that it is very difficult to understand what is happening. But with fusion proteins, you can actually try to model it. So how do people try to understand if the fusion protein itself is driving the cancer? Is to model the cancer in other ways. For example, in mice, so try to put it in a similar kind of cell, and then try to induce the cancer in mice and see if this is what is actually driving the cancer. But it is a very difficult question to answer, because it involves a lot of other parameters. Yeah.
All right. One last question. I think then we can go on to other items in this session here. So I know you did say something about you're raising antibodies to certain fusion proteins, and then And then, you'll be able to see how quickly or how efficiently our own immune system is able to detect before the researchers detect. So I think you were saying something about that, but I got distracted because I was trying to see if one of your dad's friends was actually tuned in or not. So can you, how the role of the immune system, can you answer quickly? Will the immune system, can it be enhanced in such a way that obviously the fusion proteins are foreign proteins to our system? So immune system should be up and ready to detect it and do something about it so that it doesn't manifest into a cancerous growth.
Yeah, so what I was talking to you about was actually engineering it in outside the body. So in the body, I maybe it is a case. I don't really know, but I know that targeting the fusion itself has not gone that well. Like people have tried to target the fusion protein with chemicals or through whatever antibodies, and I don't think that's such a viable strategy. That's why we are trying to study what the fusion protein interacts with. For example, an entire complex, then you have more hits to work with. So you can target the entire complex or the activity of a certain complex to treat the cancer. So that is the rationality. Yeah.
All right. Thank you, Deepthi. I think we should move on as we are running out of time. I think, again, let me congratulate you for an amazing project that you're on the lab that you're working in. I think you're getting fantastic help from your colleagues and your mentor. We would like to thank them also for allowing you to discuss your results, at least some of them. So hopefully we will connect back to you sometime in the near future, hopefully coinciding with the awarding of a PhD degree itself, so that we can have a much bigger celebration and invite more people, so that you would have made some breakthrough of how chromosomal translocations can actually cause cancer. So now I would like to go back to the organizers to see if we have your dad's friends to reminisce, some of the moments that they would have had with late Sri L. Sudarshan Gowda. So Sweta, what's next? Can you please bring somebody in? Can I bring Dr. Sheshashai, who was, I believe, his student, or he was his junior, Mr. Sudarshan Jr., and he's currently the head of the Department of Crop Physiology. And Shesh, can you please quickly reminisce what, I know it's been a while, but I'm sure he comes to your mind of, quite often. So please kindly share what you thought of, your friend and your teacher, your senior, whatever it may be, that you can bring back. Thank you, Shesh.
Thanks, Jags. And before I say anything about our Sudarshan, congratulations, Deepthi. It was an amazing thing. And when Jags asked, do we have any questions? I was actually appreciating what she was saying rather than to find out what questions we can ask. And my best wishes to her. And I remember her as a very, very small child. She used to come and she was telling that she used to come to GKVK, and biochemistry lab was just below our physiology department. And biochemistry used to be a very powerful lab those days, with Sudarshan, and Virupaksha was the head of the department, was a professor then. And there was a very powerful team. I remember this I was a student, I should say, that is how it was. And then, those days, though he did not teach me in a theory class, he used to come for the practical classes, and it was amazing. Practical classes are something which was very, very important for us in those formative years. It was in 1985 when I was a master's student. And, there was something that I especially remember is that we used to specifically discuss about nitrogen metabolism. That was something which was very dear to us, because I was working on carbon assimilation, and then they were very, very good in understanding what the nitrogen assimilation was all about. And now we have, I should say that the kind of discussion that I had with Sudarshan those days, after several years, 2-3 decades, we have a program on understanding how these two can be linked. There is something that we are trying to now improve is the protein. If you increase the protein content, which happens by the nitrogen assimilation, then the carbon assimilation has some kind of a trade-off. We were discussing about those things, and I was wondering how cruel the damn nature is that it has to take him away so early. Now, one of the most exciting thing that happened to me with Sudarshan was we used to have this Friday groups and Tuesday meetings, and where we discuss science. And I was mostly interested in sports those days. But then when it comes to the science, when we discussed with Sudarshan Jagannath from veterinary science, and then we have Uma Shankar, Ganesh Aya, all these guys. We had a real nice time listening to those seniors who were so exuberant with energy and enthusiasm and so much of knowledge. But what happened in around 2001 or something, maybe early 2002, we had a major research grant. That was, I should say, was one of my major fundings that I received from the Department of Science and Technology and Department of Biotechnology, was to establish a national facility here in GKVK, which runs even now, right behind me, behind this wall. And that's about a mass spectrometer. And we had two major things to resolve in that project, was to look at how physiology can help improve drought tolerance, and how biochemical processes help these different processes. And that is when Sudarshan was a investigator alongside me. He used to always tell me, it's amazing to work with my student, shoulder to shoulder. And that was something that now there is very less time. Of course, I know that. I appreciate that a lot of other people would like to now reminisce and then tell good things about it. What Deepthi did is actually reminds me of what I did with Sudarshan. And therefore, it's something similar to that. There is one saying in Kannada, Maga Tandena Mirisbeko, Shishya Guruna Mirisbeko, Agle Gurugu Tandegu Tripti. And that is something which is very beautiful. I'm very sure Sudarshan, wherever he is looking at us and then happy. He's feeling very happy that Deepthi has done so very well. And then I'm really happy that it was an amazing program, Jags. And Padmini did call me and tell me that we should do this. And it was something which is very good. And as he said, we should have it more frequently. And this is some of the very, very simple ways and a very effective way that we can actually enthuse our young people to do things better. Because I remember that Sudarshan himself told me that it feels very good to work with you. I was a student. I was not a very good student, let me tell you. I was naughty. But then it was something, at the end of the day, it was amazing. And he's passing away. I have never forgotten. Even every day I remember, because I remember this. There was a fruit that came in the way of the brake on him, and that was the reason which resulted in that accident. But whenever I drive my car, I ensure that even a drinking water bottle is not there. And I keep telling them, remember this. So please, I really have a great regard to Dr. Sudarshan. We used to call him Sudhi. When we went to the meetings of our Friday groups and science group meetings and all that. So it's amazing to have this day. Deepthi, congratulations, beta. I'm very, very happy about this. Thank you, Jax. Thanks for this opportunity.
You're welcome, Shesh. Words very well expressed. So let's move on to, I'm not sure if Honorable Vice Chancellor Dr. Rajendra Prasad has joined. If he has not joined, let's move from Bangalore GKVK to St. Louis to Dr. Ganesh Kumar, who I'm sure was also a student of Dr. Sudarshan, and he's probably come to know the whole family, as Ganesh worked in Dr. Padmini Sudarshan's team while we were all at Monsanto Research Center. Ganesh, over to you.
Yes, Jag, thank you. Can you hear me?
Yeah.
Yeah. Thanks for the opportunity, Jag. So I should say I'm uniquely fortunate for having been associated with this wonderful couple, Dr. Sudarshan and Dr. Padmini. Sudarshan was my biochemistry teacher at UAS Bangalore, and Padmini was my first manager at Monsanto. They both played a significant role in shaping my academic and professional career, respectively. It was around, I think, in 1995, my PhD advisor, Professor Uday Kumar, introduced me to Dr. Sudarshan, and he encouraged me to take as many biochemistry courses as possible. Because in physiology, we learn about whole plant science, but to understand the whole plant physiology, you need to have a strong biochemistry background. So that was the reason behind Professor was pushing us to take more biochemistry courses. But it was a bit nervous in the beginning for me, mainly because the general perception was that in those days it is hard to secure a good grade in biochemistry. So whereas Dr. Sudarshan actually changed that perception by virtue of his approachable nature that allowed students to interact more freely. So they say that, right, to a good learning to happen, there should be an environment has to be created. I think Sudarshan had that amazing, his personal attributes that enabled students to come freely interact and learn in his course. So ultimately, I secured a good grade and also learned a bit about enzymology. And I still remember the Michaelis Menten kinetics he taught us. So in physiology, we used to say, especially the enzyme Rubisco was so inefficient. So Shesha has to say it used to, Rubisco forgot to evolve. But we learned the reason for that through Sudarshan sir's class. So, I mean, those are some very interesting learnings. It's still vivid in my mind. In addition, Sudarshan sir has also served as an advisory committee member for many of our students in crop physiology. And during my PhD thesis writing, I had several productive interactions with him. What I would like to say is Sudarshan sir was not only a good teacher, but was also an excellent human being. His friendly and approachable nature brought out the best in us. So that's what we expect from a good teacher. I'm extremely happy today that as we are celebrating his life through a thought-provoking talk from his own daughter, Deepthi. And I thank her for this and also for continuing his legacy. And I wish her all the very best. Thanks a lot for this opportunity, Jack. Over to you.
Thank you, Ganesh. Thank you, Dr. Ganesh Kumar. Again, bringing back the complexities of biochemistry. Enzyme kinetics was something that was a nightmare for me also. But I wish I had learned the simplistic explanation by Sudarshan at that time. So I avoided biochemistry. I went more into genetics. But you spoke my words also. Thank you so much.
Yeah.
So now, if I'm appearing dark because the electricity has gone off in Jakkur, heavy rains here. But I'm not trying to hide myself. But hopefully, thank God to the storage battery of the laptop, I can still work through my laptop. So thank you again. So now I would like to invite Dr. Ram Mohan, who was also Ganesh's contemporary and who probably in the same years as Ganesh did, and I'm sure he had a special interaction with the Sudarshan. Ramu, over to you.
Hi, good evening or good morning. Can you hear me okay?
Go ahead, please.
Hello?
Ramu, I can hear you.
Okay, thank you. First of all, thank you very much for the opportunity, Jag and Team Biotechnica, for this. And congratulations, Deepthi, for living your dream and completing your doctoral degree in biochemistry. Let me share a few memories of Sudarshan sir here. My first memories of Sudarshan sir must be in mid-80s in the Science Forum or Science Club, I think, which Dr. Shesha already alluded to. As a raw student coming from a rural background with difficulty in spoken English, only motivation for me to get into Science Forum was this buy two coffee and occasional masala dosa being sponsored by seniors like Dr. Jagannath, Umashankar, Ganesaya, and Dr. Sudarshan was one of the constant sponsors on this. In these forum, what I witnessed Sudarshan was he was such a passionate speaker, and he argued with others in a very mild but very assertive manner. And to me, this is still the best examples. He is the best example of agree to disagree. I haven't seen anyone better than Sudarshan in that aspect. And of course, needless to say, this forum helped many of us to shed our inhibitions and allowed us to express ourselves very well. Though Sudarshan didn't teach me any formal courses, the learning which I got from him in this forum has been very valuable. Another instance which comes to my mind is when Padmini and Sudarshan, both of them visited our family in my hometown, Kollegal. This was a big group of teachers and friends had come to Kollegal to attend a wedding. And since there was time, I requested them, and a bunch of 40 plus people landed up in my home. And of course, my mother was very happy to receive them and treat them. And then Sudarshan and Padmini were newlywed at that time. And Padmini sang a few songs that time. And when both Sudarshan and Padmini sought her blessings, my mom said, she blessed them and said, Romba nalla payam, meaning in Tamil, meaning you have got a good boy, very good boy, that is Sudarshan, because she was so impressed with his mild mannerisms. And Padmini and Sudarshan are very common names in Iyengar family, and my mother just assumed that they are from Iyengars and conversed and blessed them in Tamil. But it's not easy to get appreciation from my mother. I know that for a fact. I reconnected with Sudarshan at around in 1996, 97 perhaps. That's when he had returned from UK, and when I was working in Department of Crop Physiology with Professor Uday Kumar. And Professor Sudarshan was one of a constant resource person to go to for many of the brainstorm meetings which Professor MUK hosted. That also coincided with the development of the blueprint for the Department of Biotechnology in US, Bangalore. At this period, Sudarshan's family resided in the staff quarters near BSNH College, which is now Agriculture College. And it was a very common sight to see Sudarshan sir taking both Deepthi and Kirtana dressed up in their uniforms, the white uniform, to school on his scooter. I vividly remember a bunch of senior students, especially girls, would marvel at the care in which Sudarshan sir would ready the kids for their with their uniforms, lunch box, and bags. Yes, Sudarshan left us much early before he could accomplish more. But however, he has left with a very strong legacy. And it's heartening to see Deepthi accomplishing higher degree in biochemistry. And I know Kirtana is pursuing her studies in music, another love of Sudarshan sir. And these are a few things which I want to share. Thank you very much again to the opportunity, Jag and Biotechnica. And I'm sure we will probably have more of this coming in the coming years. Thank you very much again.
Thank you, Ramu. The situation here in my room is a little bit better. I'm using cell phone lights because the current has not come back. It looks as if I'm speaking from space, but I'm still very much here. So sorry about that. Now, I think VC may not have joined yet. So we actually have recorded videos of two other dear friends, I guess students also, of Sudarshan. First one is from Dr. Jagannatha, who actually is in the east coast of the USFA. So Shweta, can you please kindly play Dr. Jagannatha's video?
A few words
Thank you.
On my dear friend. My acquaintance with Professor L. Sudarshan dates back to the early 80s, during which we both were young instructors at the University of Agricultural Sciences, whom we called Sudhi. He was in the biochemistry department in GKVK, and I was in the Department of Pathology in Veterinary College, Hebbal campus. I got introduced to him in one of the weekly seminars held by the Science Forum in the Agricultural College. Besides our academic interactions, we were also very much involved in local trekking or hiking trips during the weekends. Often, these trekking trips were field trips arranged by Professor C.K. Suresh from Agriculture Microbiology, Professor K.N. Ganeshaya from Plant Genetics and Breeding, and Professor R. Umashankar from Crop Physiology departments. As avid lovers of nature, we hiked in most parts of Western Ghats, Yana in Sagar Taluk, Shimoga district, to the Silent Valley in Palakkad district, Kerala. Pretty much every other jungle in between. In the Science Forum, we often invited experts from different disciplines to deliver seminars, followed by a Q&A session to foster scientific curiosity amongst undergrads and to broaden the horizon for master students. Whenever external speakers were unavailable, each of us prepared a topic of general interest and delivered it to participants of the Science Forum. My friend Sudhi often picked up cutting-edge topics in molecular biology. To give some examples, DNA sequencing methodologies, gel electrophoresis, reverse transcriptase, Taq polymerases, etc. Some of you may be wondering about these topics. In that context, I like to quote a title of a paper to all of you so that you can get a chronological or temporal sense of our period. The title goes like this: Primer-directed enzymatic amplification of DNA with a thermostable DNA polymerase. Psyche RK et al. Science in 1988, volume 239. So this paper got published in Science in 1988. By the way, this was the seminal paper on the PCR technique. In other words, we discussed all these topics in the Hebal campus empty lecture halls on Saturday afternoons, and I call it the pre-PCR era, where hardly we didn't know much about the genome at all, and some of the techniques in molecular biology, what you guys call a routine approach. We often sought his comments on these papers, and he was kind enough to describe at least the theoretical concepts. For me, he was not just a friend, he was my teacher too. In return, I shared my knowledge of pathology, immunopathology, and immunology in the Science Forum. I still remember we were planning on writing a grant related to the comparative aspect of lens proteins. Well, around that time, I moved to the USA. I end this presentation by thanking profusely Padmini Sudarshan and Jagdish Mittur for this opportunity to share a few words on my dear friend. And I thought of this quote from an unknown writer. There are friends, there is family, and then there are friends that become family. No doubt, Sudhi is one of those rare gems. Stay safe. I wish you all happy Dashara. Bye for now.
Thank you. Thank you so much, Dr. Jagannatha. So I think that's a very nice ending of your briefing of your memories of Mr. Sudarshan. So hopefully when you come to India next time, to Bangalore next time, I think we should all have a get-together of Sudarshan's friends and colleagues and students, and we should really discuss more about some of the wonderful interactions that you all would have had. Now I would like to invite Dr. Shantala, who also was a student of Sudarshan, and it so happened that she actually came to join Monsanto Research Center, and they probably would have worked with Padmini in a new program called Vegetative Knowledge Enhanced Program. So Shweta, kindly play Shantala's video, please. Thank you.
Good evening, everyone. With a big salute to my beloved teacher Sudarshan L, I would like to thank Biotechnica Group for arranging this webinar and also giving me the opportunity to speak about my feelings towards him. According to me, our teacher was smart looking, enthusiastic, lively, simple, and a good human being. Today I would like to share 3 thoughts about him. Firstly, he was a very good and dedicated biochemistry teacher. He used to take biochemical techniques 1 and 2, which are core practical classes. In each class, he used to stress the importance of biochemical calculations to carry out the research. And also he asked to own the responsibility while handling equipments, instruments or chemicals wherever we work. He taught us the importance of publishing the research articles and also presenting the same in various conferences. He was the second one in our university to start the work on molecular markers in 1997, which he had learned from UK. He had two projects on fishes and finger millet about molecular markers. I was so lucky to work in both the projects along with him so that I learned many skills and also I was awarded a PhD degree for working on finger millet. Secondly, he did not know to say no to anyone. He was always in the mood of sharing knowledge to others. Many MSc and PhD students from various departments used to come to him to discuss their work after 4 pm, which used to get extended till 6 pm. Due to this, his young daughters Deepthi and Kirtana had to wait for a long time. Thirdly, I want to share his dreams. He was highly passionate and had many ideas and dreams for establishing a well equipped protein lab and molecular marker lab. He also had dreams of seeing his students to work in various research institutes, MNCs, pharma industries throughout the world. When our university decided to merge our department with biotech, he was very upset. I remember on 5th December 2002, he attended a marathon meeting with VC and board members where he literally argued and convinced everyone why he needed the department to be separate. At 5:30 p.m., he came with a big smile and thumbs up. We were all so happy to see him. But God did not support for all his dreams come true. Now I am so happy that his name has been made eternal with our department at USGKVK Bangalore by Mrs. Padmini Sudarshan. She has sponsored a gold medal in his name for the best student of the department. Again, I salute to both of them. Thank you.
Thank you. Thank you, Shantala. Very appropriate indeed. You used a word eternal in your last sentence. That reminds me of a famous quote from one of my favorite movies, Gladiator. It goes like this: what you do in this life echoes in eternity. Uh, so I think, the reminiscence of all of you, students, colleagues, and friends of Sudarshana clearly reflects the role that he would have played and how it will continue to echo for generations to come through his legacy, which will be his daughters and probably grandchildren as we move on. So I'm not sure. Sweta has VC joined. I know he's been very busy. He's been traveling from the airport to Belagaum and places like that. I'm not sure if he's able to catch this session. If he's not there, then I think I just would like to end this part of the event before I pass it on to Sweta and Biotechnica CEO Shekhar Suman. I just want to thank all the friends who've come forward to share their memories of Mr. Sudarshana. I would like to thank the family who has been very supportive. It was not their idea, it was our idea, friends of Sudarshan and friends of Padmini, to make sure that we do remember him in an appropriate manner. And I think sincere thanks to Deepthi, because she's the primary speaker and she would have really worked very hard to get that talk going. And I know it would have been emotional as well, but I think she was a champion in terms of putting that aside and actually discussing cancer, what leads to one of the reasons that can cause cancer. I must also mention that Deepthi was so sure of pursuing her career in this area of biological sciences. She even made sure that the partner that she chose to be her husband, also from the same area. So Samarth, I'm sure, is listening to it. Samarth is a bioinformaticist, and so together Samarth and Deepthi would make, I'm sure, wonders as they both pursue supporting each other to find answers to this dreaded disease called cancer. So with that, I would like to end my part here and pass it back to the organizers, Sweta and Shekhar. Thank you so much.
So sorry to disturb. There is a small audio issue. You're not audible. No.
Hello. Is it audible now?
Yes, sir.
Okay. Probably there was an issue with my mic. Right. So I'll just take 2 minutes. So the picture which you are seeing on your screen right now, that's Sudarshan sir with his family and the small daughter which you see there. She is Deepthi. And all of you, thank you so much for attending this webinar. Today, we didn't just celebrate legendary teacher, we also celebrated and learned a lot.
Sir, sorry, you're breaking up, sir, the audio.
So with this, I would like to conclude the webinar. Thank you everybody for attending this session. It was really, really wonderful talking to all of you. Thank you.